relate that to either the growth or the yield. And the general response is that you get this initial rapid increase. So when the plants are showing severe deficiency, small increases in nutrient uptake will result in large increases in growth. But as you approach the optimum level, that will slow down, and there's quite a wide range of nutrient levels within the plant over which you won't get any significant change in the growth or the yield of the plant. And if you go to extreme cases, you can run into toxicity. The symptoms you see, the visual symptoms of deficiency, are generally expressed in this severely deficient zone. Once you get up into this stage here, a lot of those visual symptoms of deficiency disappear, and basically all you're really seeing is a slight reduction in growth. We call that the hidden hunger. In other words, you're getting a reduction in the growth of the plant without actually seeing any visible symptoms of nutrient deficiency. And so you may, in some cases, have a yield reduction of 5-10% without actually knowing that that's occurring. The other thing is that just simply increasing the concentration of nutrients in the plant above this critical range is actually not going to benefit the plant whatsoever. And basically what you're trying to do is aim the nutrient levels around that, for the most efficient use of nutrients, around that level. So, because the talk is on micronutrients, I thought I'd better quickly go through some of the symptoms and the effects of some of the three major micronutrient deficiencies that we can find in this part of the world. So, the first one is zinc deficiency, which arguably is the most widespread micronutrient deficiency through South Australia and parts of Western Victoria. And the symptoms of zinc deficiency generally show up around tillering and early stem elongation in the mid-leaves. And the characteristic feature of early zinc deficiency are these parallel lines of yellowing, pale green to yellowing tissue on either side of the midrib. And if that becomes more severe, you get the whole leaf dying. And you can see here the midleaf can start to collapse. It's generally quite patchy, so you can see there are weak areas of the crop in here which are showing zinc deficiency symptoms, where there are other areas which aren't. And this is probably a reflection of the variation in the soil type. We call this effect tramlining because of those parallel yellow and necrotic spots. The other thing is that zinc deficiency in a more subtle way can actually reduce stem elongation, so the plants become a little bit shorter, and the leaves, as they're appearing, are actually much more closely bunched together. And if you get zinc deficiency developing later during stem elongation and into flowering, it can actually cause poor anther development and reduce seed set. And if that's occurring, you actually may not notice it if those symptoms don't appear. We've also found, for example, that zinc deficiency improved levels of zinc can also help to protect plants from things like heat stress. They're involved in some of the protective enzymes associated with heat tolerance, and so having adequate levels of zinc is one way of actually improving a certain degree of tolerance against heat stress. Copper deficiency is another one which can occur from time to time. In the vegetative stage, you generally see this wither tip, and that's because the copper is involved in the lignification of the cell walls, and with copper deficiency you get weak stems and weak leaves being introduced, and you get that wither tip. But probably more critically, copper is involved in pollen development, and pollen actually has a very high requirement for copper. And so copper deficiency developing from booting into flowering can actually result in quite significant amounts of sterility in the head, even though you actually may not see any vegetative symptoms early in the growing season. So sometimes you find that copper will develop later in stem elongation, and the crop may look okay, but the heads may show a high degree of sterility. In the straw, if you have copper deficiency, you have what's called melanism, or this blackening of the stem, and you can see this rat-tailing, which is also a symptom of copper deficiency, which in some cases actually looks like frost. So there have been many cases where frost has been diagnosed as copper and copper being diagnosed as frost. So the two are somewhat difficult to distinguish from that symptom. One of the important things about copper is that copper deficiency is actually exacerbated by high nitrogen. So if you're in a marginally copper deficient situation and you apply high rates of nitrogen, or you grow a crop after a legume, you can actually push that crop into copper deficiency. And one of my earliest experience of trace elements was actually at a trial at South Wonwonda, where we actually reduced yields by growing wheat after lupins in a copper deficient soil, because the extra nitrogen from those lupins actually induced copper deficiency. The other one, which is probably fairly uncommon in Victoria, is manganese deficiency, and the symptoms of that is that the leaves go a pale yellow or pale green to yellow colour, and they become quite limp and floppy. And so you actually might have a crop which is growing in quite a moist soil, but it's actually looking water stressed. And again, it's partly because the manganese is involved in the strengthening of the cell walls, and in manganese deficiency, you get weak cells being produced. In oats, the classic symptom is grey spot or grey speck disease, and oats in particular are quite sensitive to manganese deficiency. In legumes, you often get a seed defect. So in peas, it's called marsh spot. So you can see here that you get these blackened hollows in the middle of the pea. And in lupins, you get split seededness. And what happens there is that the lupins become manganese deficient during flowering and pod set. You get poor manganese nutrition of that developing seed. And so you get the seed coat splitting, and the cotyledons being exposed in the mature seed. I talked about hidden hunger, and this is just a point in case. Here's just an experiment which is looking at the response to copper, and the blue line here is the total growth of the plant, and here is the grain yield of the plant. So we've gone from a level of copper which is not limiting, so the plant is quite happy at that level. And you can see as we go lower, obviously the yield and the grain yield and the biomass are reducing. But you can see here that the yield is actually only reducing slightly, the biomass is only being reduced slightly, but the yield is actually being reduced quite considerably. So that plant would actually look quite healthy, but you've already got maybe a 20% reduction in yield. And the reason for that is the copper deficiency is affecting the seed set within that plant. There's not obviously a micronutrient, but it's phosphorus. But again, what it shows is some of the effects of nutrition be quite subtle. And here's an effect of phosphorus on the growth of wheat and barley. And the point that I just want to make, here we have a wheat plant or a variety of wheat grown without phosphorus, and here grown without phosphorus. Now, without that comparison, that plant doesn't look particularly deficient. I mean, the lower leaves are slightly yellow. This is from the mallee, and in many cases, crops actually look a bit like that in parts of the South Australian mallee. But without actually comparing with that, you wouldn't say there was anything drastically wrong with that plant. But there are two things. Firstly, there is a slight